Good afternoon, everyone. Thanks for making it through this weather to hear a little bit about kidneyomics. Do we have any Duke or Carolina folks in the room right now? I'm a Duke grad. Tonight's the big Duke-Carolina game, which makes this the second most important part of my day. So I want to spend some time today and cover the following objectives. One, I really want to share with you all the frustrations that I have as a clinician of caring for patients with nephrotic syndrome. And I want to talk about the rationale that has driven me from the start to try to use genomics-driven discovery to discover treatments and cures. I'll give some examples of that. And finally, I want to tell you about the newest initiative coming out of the U of M, the Big Bird study. Here's a picture of Mikey. His mom gave me the opportunity to share this with the group, and he's a patient with nephrotic syndrome for whom I care. So this whole pursuit of started with me taking care of a patient, and Jermaine was one of those patients. He showed up in my clinic in 2009, and he presented with edema. You can see this around his eyes here. His cheeks were swollen. He had protein in his urine, low albumin levels in his blood, and he had high cholesterol. And it was a pretty obvious clinical diagnosis of nephrotic syndrome. You can see that the loss of protein in his urine led to low oncotic pressure and accumulation of fluid in his abdomen. This hurt, caused massive amounts of diarrhea, and was a source of infection. So his mother's questions to me were very insightful, as most mothers' questions are. She said to me, Dr. Sampson, why did this happen? And I said, I don't know. And she said, what's going to happen? And I said, I don't know. And finally she said, can you help him? And I said, yes, but I knew that in trying to help him with the medications that we use now, we will certainly be causing a ton of side effects, and we are only hoping for some cures with these therapies. So given that all y'all are not nephrologists as far as I know, I want to give you a little primer on nephrotic syndrome. So this is the kidney. Most of us have two, 500 of us have one. And inside each kidney, there are 1 million filtering units, or glomeruli. And what the glomeruli do is they receive blood that has waste and extra volume in it from the heart and the aorta and the renal arteries. And the blood is filtered through these capillary loops, and waste and extra fluid goes across these capillary walls, and that forms the urine. Clean blood with less fluid then returns to the body. So we each have about 1 million of these glomeruli in each kidney. Now these glomeruli, this is a scanning electron microscopy image, shows here the capillary loops. This is one of these blood vessels, and surrounding the blood vessels are my favorite cell in the body, the podocyte. And what you can see is this fine interdigitation of these foot processes, which literally hug the glomerular capillary loop, helping to prevent good products from leaving and providing a barrier. It's abnormalities of the podocyte that lead to nephrotic syndrome for the most part. This is another electron micrograph of the glomerular capillary loops with the podocytes. These are the cell bodies, and you can see the beautiful organized architecture that's serving the function. And this is a healthy example. And this is what the podocyte and the glomerulus looks like in nephrotic syndrome. You can see that there's a loss of architecture, a flattening of these foot processes, and the general idea that it looks like some sort of bomb went off in the glomerulus. And this is what leads to the nephrotic syndrome. No, when it hits, whether it's adult or pediatric. And we don't know yet if the etiology is the same in adults or pediatric. We know that there's some differences, but we know that when we see a patient like Jermaine, we know for sure that his foot processes look like that. So here's the current lay of the land in terms of the classification of nephrotic syndrome. And what I want to emphasize is that it's descriptive and imprecise. For patients who we don't biopsy, we classify them based on their response to the first-line therapy, which is steroids. And if a patient is sensitive to steroids, if those steroids can induce remission, then they get a diagnosis of steroid-sensitive nephrotic syndrome. And conversely, if they don't, they get a diagnosis of steroid-resistant. I talk about the cart coming before the horse, or a post hoc analysis. We should not have diagnoses that are dependent on a response to therapy, but that's what we have right now. For those individuals who get a kidney biopsy, which is a small invasive biopsy through the back, we look at the histology under light microscopy, and if the patient has minimal changes in their glomeruli, they have a diagnosis of minimal change disease. And if there are focal areas of glomerulosclerosis, then we give them the name of FSGS. That would be like calling breast cancer lump in the breast cancer, or lump in the breast syndrome, not getting to the precision underlying molecular mechanisms. These descriptions, while useful in some ways, have really limited our ability to be precise with our therapies. The other thing we have knowing is we have an inability to know exactly what's going to happen. Now, this is germane over the years, and you can see periods of doing well and not doing well. What we know is nephrotic syndrome, when this patient shows up over time, a percentage of them, the minority, will have steroid-sensitive nephrotic syndrome and will never relapse after their first remission. Others are sensitive to steroids, but are dependent on them, or they have frequent relapses, which means that we have to constantly give medications to induce remission at a cost of side effects. And some patients won't respond to any immunosuppressive medication, giving them steroid-resistant nephrotic syndrome. These patients are at risk of substantial kidney dysfunction, leading to chronic kidney disease or even end-stage kidney disease, needing a dialysis or transplant. And the heck of the situation is, even for those patients who get a transplant, a percentage of them will have a recurrence of their disease attacking the new graft. The morbidity and mortality of nephrotic syndrome comes from the nephrotic syndrome itself. There is infections, clots in the blood, in the bloodstream, in the vessels, and the quality of life is pretty poor. And we also know that the mortality and morbidity comes from the treatment of the nephrotic syndrome as well, because we've basically stolen immunomodulatory drugs from the oncologists, and we hope that we can modulate the immune system in nephrotic syndrome as well. So Jermaine got one of those treatments, which led to accelerated hypertension, seizures, coma, which has given him a long-term seizure disorder. And over the last 5 or 6 years, he has mostly been in remission, but in the last 18 months, he's returned to having the nephrotic syndrome, and we don't know why. This hits me in the head and the heart. So there are many folks who care about this just as much as I do. Many of the folks here in Ann Arbor, which is one of the reasons I came here from Penn. I've chosen to dedicate my career to addressing this lack of precision through genomic discovery, with the idea that if we understand the genetic underpinnings of nephrotic syndrome, we can get novel insights about the biology. We can use this biologic understanding to identify new targets for medication. We can discover the clinical impact for those harboring the disease variants. And in addition, if we understand a patient's underlying molecular signature, it can help us select them for particular clinical trials. And ultimately, the idea is to match patients with treatments that most likely benefit them. Now, this was not a unique insight by me. I was influenced by work done when I did a year of cancer research at Children's Hospital of Pittsburgh. And unbeknownst to me, Matthias Kretschler and Larry Holtzman and other folks here in the Department of Medicine and the CCMB at the time came together with this idea of a genomic-driven inquiry for nephrotic syndrome as well. So when I finished my fellowship training, this was a natural place for someone who had a lot of enthusiasm and energy, but no money and no clue to come and be mentored in how to do this. So genetic discovery in nephrotic syndrome is about a decade and a half old, and like most genomic discovery, started with the world of Mendelian mutations. Over the past 15 years, about 60 Mendelian forms of nephrotic syndrome have been discovered, and those have some very strong clinical correlations. And there's a lot of hope and interest and excitement about these discoveries, both for clinical correlations and also identifying mechanisms. But the fact of the matter is, Mendelian forms of nephrotic syndrome represent a minority of the genetic causes of diseases for our patients. While a recent study showed that in the US, in Ann Arbor and Los Angeles, about 13 to 14% of our patients with nephrotic syndrome have Mendelian forms of disease. In work that we did in our Neptune study, about 6%. We see in other places that it can range higher. And a lot of times the higher prevalence is due to consanguinity driving that. So the majority of our patients are not going to have a Mendelian form of disease, which creates an opportunity to take an alternative approach to discover disease-associated genetic variation through population-based approaches. This dovetails well with my training at Penn, where during the advent of SNP genotyping and genome-wide association, I was really coming up during that time. So I want to share with you the conceptual model of everything that drives our lab. And I want to represent the spectrum of genetic variation as a gumball machine, which I'm sure you guys think about all the time, of genetic variation gumballs, right? So what I view genetic architecture of nephrotic syndrome is composed of a heterogeneous group of genetic variations, whether it's structural variation or single nucleotide variation, common or rare. And the number one goal of our group is to use computational medicine, biostatistics, genomics to discover known or novel loci associated with nephrotic syndrome. Because if we can do that, if we can find gene X, we can go in two different directions, okay? From a clinical epidemiologic perspective, we can contextualize our patients and stratify them as a function of this genetic risk. And we can understand the association with demographic characteristics, baseline clinical characteristics, or longitudinal outcomes. In this sense, we don't even need to understand the mechanism by which this genetic variant is associated with disease, because if this serves as an effective biomarker, it can help improve our precision of care. At the same time, however, I'm not satisfied with just doing that. We want to understand why. So what we can also do in parallel is take these genetic variants and try to define the molecular mechanisms through integration with other genome-wide data sets, through work in functional systems, and in doing so, identify targets and discover mechanisms. So we're ambitious, and I have 40 years to do this, so we want to try to attack all of this. And one of the keys to attack all this is do this in collaboration, because not one person can do all this ourselves, and I'm too extroverted to try to do it all by myself. So I want to share a couple stories that have used this conceptual model to work on some gene discovery. So I want to talk about APOL1 associated nephrotic syndrome, as well as some EQTL studies. And the other point I want to make is to tell you that a lot of these discoveries have really been empowered by the existence of a living laboratory that was started here by Matthias Kretzler and others, called the Nephrotic Syndrome Study Network, or Neptune. So what Neptune is it's a longitudinal observational cohort that recruits patients in multiple centers around the US, and because there's one site in Canada, we could call it an international study. And these patients are recruited at the time of a clinically indicated biopsy for suspicion of a form of nephrotic syndrome, minimal change disease, membranous nephropathy, or FSGS. At this point, there's more than 650 or 700 patients recruited. There's a lot of variability in these patients who are recruited. They're of diverse ages and ancestries. They're not selected on the basis of immunosuppression. So a lot of other genetic studies have been a selection. There's been a lot of selection bias, whereas this is not the case. And because these patients are followed longitudinally, there's an opportunity to really get a better understanding of what's going on. In addition, there is a collection of blood, urine, and there's whole genome sequencing that we've done on about 650 patients. RNA-seq of kidney tissue that's been led by folks in Matthias' lab, and Felix Eichinger and V.D. Nair, who are here today. And there's also serial and blood and urine samples as well. So this creates the opportunity to do stacking of omics data sets and clinical data to try to tell a story. So I'll tell you one. So let's talk about APOL1. So for decades, we've recognized that there are racial disparities in kidney disease, that while black individuals make up 13% of the US population, they make up a third of those individuals who have end-stage renal disease. Black individuals have a 4 times increased risk of getting FSGS, the more severe form of nephrotic syndrome. And for those children with FSGS, black individuals have a faster progression faster time to progression to end-stage renal disease, as well as a higher chance of developing end-stage kidney disease. And for decades, folks have used very judgmental and ultimately false explanations to explain these disparities, when there's a recognition now that there is a molecular and genetic underpinning for this. That's APOL1. So APOL1, or apolipoprotein L1, has now been discovered to be a major component of nephrotic syndrome in black individuals. APOL1 has 2 common exonic variants, G1 and G2, which are strongly associated with kidney disease. So black individuals who have a high-risk APOL1 genotype, or 2 risk alleles, have about a 15 to 20 times higher risk of getting kidney disease than those with 0 or 1. And this is a common risk allele in those of African ancestry. And for those of y'all who recall the story of sickle cell disease and malaria, it's the same story of positive selection because of the host defense system. So about 8,000 years ago in Africa, people were dying of trypanosomal disease, or sleeping sickness. Apolipoprotein L1, which circulates in the body in the trypanosomal lytic factor, developed a genetic variant. And individuals who had one of these genetic variants were less prone to die of trypanosomal disease. And because of this, there's strong positive selection. It became pervasive in the population. But over time, this advantageous allele has been shown to cause kidney disease for those with 2 alleles. So what that means for us nephrologists if this is a representation of all the black individuals with nephrotic syndrome that we see in our clinic, 70% of those black individuals, FSGS or nephrotic syndrome is going to be due to the APOL1 high-risk genotype. And that's really exciting to me and a lot of other folks, because it means if we can get to the bottom of this, the molecular mechanisms, we have the opportunity to help and treat a lot of individuals. So one of the first studies that we really undertook in the Neptune Project, in our lab with Matthias's lab, together, we really studied APOL1 nephropathy in the Neptune cohort. And what we did was pretty simplistic. We took 90 African Americans, of which about half of them were black, and we simply stratified them into those who had the low-risk genotype in green up here versus the high-risk genotype. And once we did that, we then did epidemiologic studies and mechanistic studies, taking advantage of the transcriptomic data that's been generated from these Neptune patients. We ultimately then took advantage of another independent cohort of children with chronic kidney disease that have been collected in America as well. And some of our major questions, which I'll go through briefly here, are what's the clinical impact of the high-risk APOL1 genotype for our patients who have it? What are factors modifying its penetrance, and what is the molecular mechanism? So in a series of papers that were published a number of years ago, we compared the individuals with high versus low-risk genotype. And what we found is compared to individuals with low-risk genotype, those with the high-risk genotype have a 17 mil per minute decreased kidney function at presentation. And 17 mils is a big deal, all right? So all of you, most of y'all are not nephrologists in the room, but take my word for it, 17 is massively lower. On a kidney biopsy, and these biopsies were done at presentation, individuals with a high-risk genotype had significantly increased inflammation and fibrosis and tubular atrophy. Those children who had the high-risk genotype, they had a faster decline of eGFR over time, and they had about a third chance of responding to the commonly used medications that we give to them. So what this told us is that these individuals with a high-risk genotype are a sicker group, and we need to pay closer attention to them. And we had hints with the inflammation and the lower eGFR presentation that the horse is out of the barn before we even before we see them. So at initial presentation, these patients are scarred and they're inflamed, which really prompts us to try to figure out how we can maybe pre-symptomatically diagnose them. Another aspect that we really wondered about was based on our understanding that there's incomplete penetrance of the kidney phenotype among those with the high-risk genotype. So 13% of the African American population has a high-risk APOL1 genotype, but each one of those individuals has only about a 4 or 5% lifetime risk of developing the disease. So we wondered, what are those factors that potentiate the penetrance, or those factors that protect? And the design of the Neptune study with deep phenotypic data helped us to figure this out. It was a discovery in Neptune that was replicated in CKID. This was Cassie, who was in our group a couple years ago. She's now getting her PhD at UVA, and Derek Ng, an epidemiologist with CKID. And what we noticed is we noticed that black individuals with a high-risk APOL1 genotype had a significantly greater odds of being born prematurely. So what that implied, what that specifically said, is that if we have a black patient in clinic who has kidney disease and a high-risk genotype, there's about a 5 or 10 times increased risk that they are born prematurely. What we know is that APOL1 is linked to kidney disease, and we know that prematurity is linked to kidney disease. We also know that there are racial disparities in prematurity in the African American population. So we wondered what the relationship was amongst these factors. In data that I'm not going to share right now, but which we published, we found that APOL1 genotype was not directly associated with prematurity, but we didn't have power to discover whether APOL1 genotype was associated with poor birth outcomes. So what we think is going on right now is that if this is the baseline risk for FSGS or kidney disease in the population, and this is the threshold to develop disease, that having the high-risk APOL1 genotype increases the risk of getting over that threshold. Being born prematurely increases the risk of getting over that threshold, and those two factors together tip over. What was interesting and somewhat gratifying is that after publishing this, in quick succession over the past 2 years, there's been more evidence that APOL1 has a role in birth outcomes. So in a study of APOL1 transgenic mice done by our colleagues and collaborators in Cleveland, they were trying to overexpress APOL1 in mice, which don't ordinarily express it, to identify kidney disease. While they didn't really find a lot of kidney disease in these mice, they did find that these maternal, mouse moms developed preeclampsia and had preterm birth and problems. In addition, a recent paper did epidemiologic work, and they looked at APOL1 genotypes of pregnant mothers and then their fetuses, and they recognized that fetal APOL1 genotype is associated with risk for preeclampsia. So we now have a pathway to pursue to understand even more whether this APOL1 genotype in the infant sets off some reaction on the maternal side. We know APOL1 is strongly expressed in the placenta, and whether that is ultimately something that we need to attack. Because this could be really important if in fact that premature babies who have a high-risk APOL1 genotype have a high risk of ultimately developing kidney disease, then we could work on community screening and identification of these children, perhaps do more screening at this point. And when APOL1 associated medicines are discovered, we may be able to target them as well. The last question that we really want to address is what are the molecular mechanisms underlying this? So what we did is a genome-wide differential mRNA expression in the glomeruli of Neptune patients with a high risk and low risk APOL1 genotype. It was such a unique opportunity to have the actual kidney tissue, which is the target of the disease, rather than relying on circulating blood transcriptomes, etc. So a volcano plot was made, and what we have on the x-axis is increased expression in high-risk patients, decreased expression, and this is level of significance. In red, each dot is a gene, and with a very small sample size, we were able to identify 3 genes that were significantly increased in the expression of the high-risk genotype. The 2 chemokine receptors and UBD, or ubiquitin D, which ubiquinates or tags proteins for degradation. So we wondered, each of these is targets, and as we increase our sample size, more of these are going to be significant, and we wondered what the role of these are in disease. So when this happened, I called up Martin Pollack, one of the guys who discovered APOL1 as a kidney risk gene, and asked him to look at UBD, and in collaboration with David Friedman, and we contributed a little bit, we followed up and published this paper that UBD does in fact modify risk. And what they did was 3 studies. They did one study where they looked at African admixture among African American patients with kidney disease, with other forms of disease, and healthy controls. And what they found is genome-wide, the UBD locus was strongly enriched for African ancestry by mapping through admixture linkage disequilibrium. Then they took cell lines from the HapMap project and compared UBD expression among African individuals and European individuals. And they found that African individuals had a lower expression of UBD. And finally, they did studies in cell systems where they overexpressed APOL1, which caused the cells to die. But then they overexpressed UBD in these cells, and there's less cell death because of UBD. So the hypothesis now is that UBD is protective against APOL1-induced kidney damage, and black individuals, because of genetic factors, have a lower expression of UBD. So moving forward, this is one target that we can go to depth on while we continue to discover more molecules. And again, an example of how collaboration, both within the Neptune group in our labs and external groups, can really move this field forward. The other major story I want to talk about is of EQTL studies and our ability to do EQTL discovery in the Neptune cohort. So most of you all in the room probably know what an EQTL is, but just for those who might not, an EQTL is the concept that there is a genetic variants that influence gene expression of usually genes nearby. That would be a cis EQTL, likely via regulatory effects. Doing EQTL studies gives us sort of an agnostic look at what genetic variation can regulate gene expression. Now, EQTL studies are interesting for a bazillion reasons, but I'll just tell you 2 broad reasons. One is that EQTLs may be able to help us understand the links that we discovered through genome-wide association. So this is a representative of a genome-wide association or a Mendelian disease where you have a nucleotide change that ultimately results in a phenotype, a clinical phenotype. I really want to focus on complex traits and SNVs that are this relationship is discovered via GWAS. It's not, because most of these are not exonic changes, we're left to hypothesize how this is linked. Well, with EQTL studies, we can link a genetic change, a GWAS SNP, to gene expression, and then we can link that gene expression to outcome. So we can try to create a causal relationship. Another example of why EQTLs are important is it helps if we begin a study by identifying EQTLs, we can reduce the search space, which is quite punitive when we do genome-wide association studies. So as many of you all know, when we do a genome-wide association study, which is a major effort that our group does, which I'm not going to share today, we have to use a p-value of 5 × 10 to the negative 8, and an association must surpass that threshold to be considered genome-wide significant. It's a pretty conservative estimate, so we know that there are likely a lot of false negatives. So instead of doing a million tests, if we start by discovering EQTLs, we can then study the association of these EQTLs with outcomes and not have this punitive of a p-value threshold. And in addition, we are starting our analysis with genetic variants that we know impact the expression of genes in the cell types or tissue types we care about. So there's been a lot, a lot of this work has, and a lot of the, there's been a lot published on EQTLs outside of kidney disease over the past number of years, and it's been really fun to go through the literature to understand what folks are finding. So one thing that has been identified over the years is when GWAS studies are done, whether it's type 2 diabetes, height, hypertension, lipids, these GWAS alleles are enriched for EQTLs in target tissues. So GWAS hits for coronary artery disease are enriched to be EQTLs in coronary artery tissue. Alzheimer's SNPs are enriched for EQTLs in brain. And for those of you who are interested, I highly recommend this paper, which was published last year, and William Wen is a co-author on this as well. It came out of GTEx. Besides enrichment in target tissues, we also recognize that about 10 to 15% of the EQTLs we discover are specific to between 1 and 3 tissues. So this gets to the point that there's an argument to be made that many EQTLs are shared among tissues, and for many cases, perhaps getting a blood sample and isolating blood cells and doing transcriptomics there can allow us to understand EQTLs just as much as getting the target tissue. But we recognize that there is specificity and importance in getting the tissue that is under disease associated. But the problem is that there are no publicly available data sets of kidney EQTLs. So this lack of these data sets, whether it's in GTEx or Juvetus or elsewhere, presents an opportunity. And in a collaboration, again, with the Kretzler Lab and William Wen, and led by Chris Gillies and Rose Putler, who were in my group recently, we put together and published an EQTL study of glomerular and tubular interstitial tissue. And it's important to point out, as much as important as is our drive to discover, I'm also highly invested in sharing and making use of this data for a wide group. So all this data is available on nephqtl.org as well. So what we did was we had already done whole genome sequencing on patients in Neptune, and work led from the Kretzler group has done Affymetrix-based array expression from the microdissected glomeruli and tubular interstitial compartments of the kidney. What I should mention is, remember I showed you the kidney at the beginning. About 85% of the kidney is made up of tubular interstitium, which does a lot of transport and has some immune functions, and only about 10 to 15% are glomeruli. They have very distinct biologic roles, and there's a risk if we did whole kidney expression that we would miss and confound associations. So work done by the Kretzler group and a lot of time-consuming laborious work, they pluck the glomeruli out of the tubular interstitium and then do paired transcriptomic data sets. So it's a massive advantage to have this. We then, led by programs that William has developed, we did matrix EQTL, torus, and DAP. These are Bayesian approaches to identify credible SNPs that are probably driving an EQTL signal. And when we did this, we discovered in our glomerular EQTL data, well, this is not the discovery, this is sample size. We had 136 glomerular samples in our sample set and 166 tubular interstitial data sets, which is larger than many tissue types in GTEx and larger than one previous kidney EQTL project. We then, once we discovered the EQTLs, we then took advantage of other multi-omics data sets for integration, including epigenomic data sets and kidney single-cell RNA-seq data sets. So that was the outline of the study. And what we ultimately identified were in 833 glomerular EQTLs and about 1,800 tubular interstitial EQTLs. And this is not only saying that we discovered an association between a SNP and a gene expression, but we were also able to pinpoint the likely SNP or group of SNPs that are driving this expression. This is the fine mapping or Bayesian approach. We then compared these GWAS SNPs with, these eSNPs to the EQTLs in the GTEx tissue. And in fact, we found that there was quite a bit of specificity. So the glomerular EQTLs, about 10% of them we would not have found if we relied on any other tissue. These were unique to the glomerular. And 14% of the tubular interstitial EQTLs were unique to that tissue, not even in the glomerulus. So those may be the most disease-specific or tissue-specific, although they're clearly tissue-specific by definition, but those may be the most high-impact allele or genes to look at for further downstream studies. We took those EQTLs and those eSNPs, and we then overlaid them with the roadmap epigenomics data set, which uses chromatin and methylation to identify enhancers, repressors, transcription start sites, et cetera, in a variety of data sets. We looked at fetal kidney, and what we identified is that our EQTLs were significantly enriched in transcription start sites and enhancers. And this is not shocking. Folks who do this would say that most eSNPs are enriched in this area. So for us, it's more of a sanity check that what we were doing was rational. So now it's cool to discover EQTLs, and we certainly featured that prominently in the paper, but it's like, this is just the starting point. What can we do with it? So it's been really cool to see that since its publication in August, there's been 4 papers that have used this in publication to annotate these EQTLs and eSNPs to annotate their work. But what we've been working on, what we've presented in this paper, is a focus on relating these EQTLs to nephrotic syndrome. So we transitioned from doing genome-wide work to focusing specifically on those 894 EQTLs or E genes seen in the glomerulus. And I wondered, if you remember from the beginning of the talk, my favorite cell is the podocyte, and the podocyte is the major cell involved in nephrotic syndrome. We wondered whether these glomerular EQTLs were enriched for podocyte genes. So to do that, we collaborated with Matthias Kretzler and Nir HaKohen at the Broad, and in analyses led by Raji Menon and Edgar Otto, they had done single-cell RNA-seq on about 4,700 kidney cells from adult tumor nephrectomy tissue. And in doing single-cell RNA-seq, they used the 10x genomics platform for this particular experiment. They clustered cells that had a similar signature, and then by identifying key genes, they were able to identify specific cell types that were associated with these clusters. There are about 15 clusters. And then what we simply did, we laid out the clusters on the x-axis here. And then each of those 894 genes, we said E genes, is that E gene specifically enriched in any of these cell types? And if it wasn't, I probably wouldn't have shown you that slide, so that's a hint that we did find something. What we see here in black bars are glomerular E genes, and in gray bars are tubular interstitial E genes. And it was incredibly exciting for us to find that glomerular E genes were most enriched in podocytes. So this gave us the idea that we could now take our glomerular E genes and dissect out those that are podocyte specific, and then further study those genes for their function. So the first thing we did was we did functional annotation using Go pathways of the 80 to 100 genes of those 894 that were podocyte enriched. And we found that the biologic processes, the molecular functions of the single transduction pathways really jived well with our prior knowledge of the biology of the podocyte, whether it's vesicular-mediated transport, locomotion, integrin signaling, etc. We were finding those genes that tag biology we knew. And we saw some of our favorite friends of these genes here, but we also saw a whole group of genes that we hadn't yet recognized may be podocyte specific or important. So with this group of genes, we have another wide variety of next steps to take. And one of the next steps we took, again, with Matthias and Raji and Jennifer Harder, we started overlaying these candidate podocyte genes with organoids. So worked on independently of our group, has been, over the past couple of years, the development of kidney organoids from induced pluripotent stem cells and fetal kidney. And for those of you who aren't totally into organoid maps, you can look at cell trajectories over time and take a group of cells that are undifferentiated that go down different pathways in time series. And we can identify those that are in the podocyte lineage, etc. And another way to look at this is static. We can look at it in a two-dimensional form where this is time on the x-axis, and each row is a podocyte specific gene. And the colors are the intensity from low expression to high expression. And what we're looking at is over time, the turning on and turning off of genes through podocyte development. So what we see in this list of genes, we see some of our known genes, VAMP8, which is a vesicular transport endolysosomal gene. We see PLA2R1 and thrombospondin, which are known genes that are the major causes of membranous nephropathy, form of nephrotic syndrome. MyH9, which is a known nephrotic syndrome gene. So we see genes that we know, and we could say that PLA2R1 is a podocyte gene that turns on late in development, which everyone has known. So that's another sanity check or positive control. But it means that for these 75 other genes here, we can start to get an idea of their role in biology. And then we can go back to the eSNPs and the EQTLs and figure out why these genetic variants are influencing their expression. And that's one direction. So we can go to genome biology, and we could think of it, but we could also go forward and saying, is the expression of these genes associated with clinical outcomes? So a lot of work done in our group, and as well as Matthias's and others, is to take gene expression to see if these gene expression differences are associated with outcomes. So a perfect story, which we've seen hints of here and there, is if we identify a gene whose increased expression is associated with badness, and then we find a genetic variant that's associated with decreased expression of that gene, then we might want to figure out what the biology of that is, because if we could give like a dose of that genetic variant, and that could decrease expression and lead to better outcomes, that would be a cool, really cool drug target. So I really view our role as from the gumball analogy, is to use really rigorous computational statistical genomics to bring forth these data and to then share them with the world, whether it's NEPTL or NEPDS or collaborations, to allow people who are organoid experts or transcriptomic experts or clinical epidemiologists to take advantage of this data, which could only be put together by a collaborative group for future discovery to ultimately impact patients like Jermaine. So some of the next steps, there's a lot to do. And like I said, I have 35 years, probably more to go here. So I think I'll have enough to do over that time. So a lot of it's about recruiting more patients. It's about generating more interesting data sets from the existing patients, and it's functional analysis. So I'll give you a couple examples. In work done with Jacob Kitzman, we're working on high throughput functional classification of variants of unknown significance. So, for example, WT1 is a known Mendelian form of nephrotic syndrome. And these days in the clinic, patients are getting sequenced all the time. And patients are being found to have variants that are definitely Mendelian forms of Mendelian mutations, as indicated by these red and blue dots. These are known genetic variants. But the majority of genetic variants that we find on testing, it's impossible to classify as being totally benign or truly pathogenic. And it matters, because we'll make clinical decisions based on this understanding. Now, it's financially and temporally impossible to make a mouse for every one of these variants to understand outcomes. So work that Jacob has developed over time is to do these programmed allelic series on a gene level. And the idea is to take a coding sequence of a gene, this is from one of his earlier publications, and for every amino acid position to create every other possible amino acid of that position. So for instance, at this residue here, each row here is a different amino acid. And then once we have a cell with that protein, which has that different amino acid, to assess via readout what the impact is. And this is showing intensity of a readout on a scale, and the darker blue means there's a bigger impact, and yellow is the wild type, and white is not. So for WT1, what we're doing, led by Jennifer Yee in our group, a postdoc, she is taking WT1, which is a known transcription factor, and we know what WT1 can bind to the promoter of nephrin. So she is perturbing every single WT1 variant and then testing its impact on turning on nephrin. And in that way, we ultimately hope to come up with maps so that we as clinicians, when we see a genetic variant, we can more accurately classify as harmful or not. And if we can do it for WT1, then the idea is to try to figure out readouts for more and more of these genes. Work that's being done to increase our sample size. This is a rare disease, so we can't just recruit patients from Michigan. It's incumbent upon us to collaborate and develop worldwide collaborations. And a group of 4 of us, Freedom Hildebrand and Martin Pollack, both at Harvard, Simone Sonikerke at Columbia, and myself, we have put together a cohort that ultimately is more than 10,000 individuals with FSGS, both adults and children. We've written program grants to the DoD and the NIH to support the aims that I'll briefly describe to you. We're going to bring these patients together and we're going to do a number of projects. In project 1, we're going to seek to identify novel Mendelian forms of FSGS in Mendelian cases. In project 2, we're going to treat nephrotic syndrome as more of a complex trait in these rare variant association studies and genome-wide association to identify genetic variants. And once we find those variants, we will then go into project 3 and take advantage of our collective expertise in model systems, whether fish, flies, cells, to functionally characterize any of the genes and variants we find. And in addition, if in the context course of doing animal-based or system-based models, if genetic variants or genes become of interest, we can then screen our genetic data to see if they matter. And finally, in concert with all of this, in project 4, we want to turn this genetic data into clinically actionable information. So we are going to test a trial of rapid next-generation sequencing to do diagnostics within 1 or 2 weeks of patients with nephrotic syndrome to see if that impacts care. Preliminary data shows that it does. And we also want to build on the NephVS and NephQTL that our group has worked on, and to create the equivalent of the exact database or NOMAD, 1,000 genomes, whichever publicly available browser, in that spirit to share this genomic data around the world. And we've already, from the traffic that's hit our websites, NephQTL and NephVS, we know that these things are being used. And it tickles me that that's the case. And in doing so, we can, by doing this, we can work on more of our GWAS. So this is a GWAS I didn't explain to you guys today. This was recently published. We found HLA and immune dysregulation to be a massive form of part of nephrotic syndrome based on this Manhattan plot. But we see suggestive loci across the genome. And as we continue to aggregate more patients, this is work being led by Michelle McNulty in our group, we are able to discuss these loci become genome-wide significant, and again, targets for us to get in more deeply. And I should mention NephQTL and NephVS is being driven by Damien Furman in our group, who's here as well, who's moving that forward. And the WT1 and the Mendelian alleles and the sequencing of patients worldwide is being led by Virginia Vega Warner, who's here as well. So finally, I want to finish with a brief description of the newest exciting thing going on in our group, the Big Bird Cohort, which stands for the Biobank to Illuminate the Genomic Basis of Pediatric Disease. And this is a logo that I made while procrastinating on service at the end of last year, made with only PowerPoint shapes. The most creative I've been in about 2 decades. So Big Bird was officially born on December 21st, 2018. What Big Bird is it's the child arm of the Precision Health Initiative. So the Precision Health Initiative has been invested in here on a university-wide level over the past 7 or 8 years. The Michigan Genomics Initiative, led by Ghazal Abbacasis, Chad Brumman, and Sachin Ketterpal, have recruited adult patients for biobanking, linking to EHR, and for discovery. No one has done kids, which drove me nuts. And I finally had, I came in all enthused in 2011 as a newly graduated fellow from a place at CHOP, which had an amazing peds biobank, and came up with the name back then, but again, I was a little ahead of my skis at that point. But over the past 2 years, the environment has shifted, and we were able to start Big Bird. So the mission statement of Big Bird is to establish one of the world's largest pediatric genomics biobanks, and to use the massive amount of genetic data generated linked to the EMR to discover the causes of disease and disability in children. And it's been a group effort. So this was one of our earliest planning meetings with folks from the Central Biorepository, folks from the MGI. Big Bird is housed in the CBR. A massive addition to our Big Bird team was Julie Balk, who's here as well, who's the program manager for Big Bird, who is actually executing this vision on a day-to-day basis, with the assistance also from Virginia, who's learned Lab Vantage and who's involved in recruitment as well. So, and we are being trained here right before Big Bird was born. So the details, eligibility criteria are children less than 18 years of age, adults who have a childhood onset condition or a condition related to a childhood onset disease, or adults who could serve as controls. So that's everyone. The enrollment sites include the outpatient clinics at Mott Children's Hospital, the inpatient floors, the intensive care units, the perioperative areas, the blood draw stations, Brighton, East Ann Arbor, and any other site. Our consent is a two-page consent with a pamphlet. It's a low-intensity consent process. They're consented to be enrolled into a biorepository and to allow a Big Bird governing committee to approve future studies without having to go back and reconsent. They also consent to be recontacted. The parents consent, or if it's a patient 18 or over. But we've had a lot of success. Julie has said to the kids, "Will you enroll your kid?" And then we say to the parents, "You're eligible too." So the parents ultimately, they can't let their kid get blood without them getting blood for the most part. So we're getting trios. We're getting trios quite rapidly. Great question, Matt. So at age 18, we get an alert from the CBR, and we have to call them to see if they opt in. If we don't hear after 2 times, we assume that they do consent to stay. So I'm a competitive person. So this is a competitive slide. So big MGI was beating us 80,000 to nothing on December 21st, but through a massive amount of effort from Julie and assistance from Virginia, in about 8 weeks, we already have 118 patients consented. So we're coming after them. And the samples we can get from Big Bird, right now we're just getting blood for DNA extraction, but we have consent. We're just limited by resources to get urine, biopsy tissue, plasma, serum, one time or over time. But the key is, to be great, Big Bird needs to engage great partners. It makes no sense to just enroll patients if this data isn't going to be used. And I think that is a unique ecosystem environment for us to really make a transformative impact for child health through genomics. So we have the opportunity to take advantage of condition experts that work here, folks that are experts in cardiovascular disease or traits, diabetes, liver disease, nephrotic syndrome. But we also have a group of experts on this campus in study design and analysis, the faculty and trainees in the DCMB and in biostatistics and in human genetics. And finally, there are folks here that are interested, involved, and experts in methods and tech development, whether it's long-read sequencing, single-cell, SciCAR, ATAC-seq, iPSCs, organoids. And finally, we have pharma and biotech that's interested, whether it's going to be for kidney-specific work or folks that are interested in getting samples for other conditions. I think the key is to really bring this out in the open and advertise and say, hey, folks, this is here. What do you guys want to work on? So last slide. So in conclusion, a large fraction of our patients with kidney disease will not have classic Mendelian forms of their condition. And we've already discovered many non-Mendelian alleles that point to outcomes and mechanisms, and there are many more to come. Many that I haven't talked about today, which are published, and many more that are going through the pipeline. And I think I'm very excited for Big Bird, because I think with Big Bird we have this unique opportunity here in Ann Arbor at the University of Michigan to push genomic-driven discovery for child health. So with that, I'd like to conclude. I'd like to thank the folks in my lab, who I think are quite incredible. I want to thank Matthias Kretzler, who I could tell who has kids in the audience. Matthias Kretzler built the Neptune study, and there are a lot of folks in the room from the Kretzler lab, and I just want to acknowledge all of y'all for really collaborating, being part of this real group effort towards treatments and cures for kidney disease. And with that, I'd be happy to take any questions. Thank you.
Okay, I'll read the first question from Gil. This is a small print. Highlight those. Oh, that's the word.
Highlight those features.
Okay, let me read it. Highlight those features of the Neptune registry, the mechanistic investigation of the role of HR-APOL1 and the EQTL strategy to reduce search space, draw upon bioinformatics expertise and special data resources. You made a beautiful case of the value of specimen and data sharing. Also, please comment on the prospects for using proteomics and phosphoproteomics in these multi-omic kidney studies?
Yeah, so proteomics is an area that is being worked on independently of our group. But as part of the Neptune study and collaborations, there's a massive amount of interest in doing proteomics. I'm going to be out of my lane by using any of the terminology, but I think SomaScan is a proteomic approach. Can someone nod or, yeah? So people are doing Soma scans from the urine and kidney biopsy tissue and blood of Neptune patients and other cohorts. And if speaking on behalf of Neptune and the other kidney cohorts that are here, and ultimately Big Bird, it's wide open for ancillary study application or discussion on how people can bring their expertise in phosphoproteomics or proteomics for additional discovery. We didn't talk about metabolomics. Metabolomics will surely play a role as well. And what we hope to do, I've taken the example of Neptune, which was built beautifully by folks here with support from the CCMB, DCMB, and the medical school to make sure that we do a great job collecting data and specimens to allow future studies from folks who have expertise. So I hope that is done both in Big Bird and Neptune.
Hi. So you mentioned in Big Bird that one of the things you're going to include is adult controls. So if my understanding is correct, the factors you're trying to pull out are things specifically that would cause disease onset in children. So wouldn't it make sense to also have as a control adults that had kidney disease onset not in childhood as a control?
Yep, 100%. You're absolutely right.
So that control set isn't just healthy adults; it's also adults who had kidney onset in their adulthood.
Yeah. So what I was showing was the eligibility criteria, and I wasn't trying to be cute, but I was trying to be as inclusive as possible. So we want to have kids, adults who have a condition related to kids, or adults who could possibly serve as a control. So that means essentially we can recruit anyone at the University of Michigan. But when we do study design, we'd sit down, we'd collaborate and think about it. If we want to ask the question, if we want to say, is the genetic architecture of nephrotic syndrome different in children versus adults, then that would be the study to do. If we want to identify shared signals between nephrotic syndrome across a lifespan, we do adults and kids versus controls. So we're just trying to make it as broad as possible.
Anyone else? Okay.
Yeah, thanks so much, Matthew. I really enjoyed your talk a lot, and it's exciting and inspiring, and congratulations on Big Bird. I mean, that's really a neat niche. I'm just wondering if you can talk a little bit about the drug adverse events. I mean, you mentioned it just in passing, and I wonder if you can expand on that a little bit, because that's somewhere, we have some specialized forms of bioinformatics that might be able to help in there.
Yeah, so an area that like you said, I didn't really touch upon deeply is independent of genetics. What we know is that a large percentage of nephrotic syndrome is due to immune dysregulation, and that using immunomodulatory medications, whether there's mycophenolate or calcineurin inhibitors, steroids, benefits some of these individuals and gets them into remission, which is what we all want. Some of those patients, in addition to getting the benefit, get side effects, and some patients don't get any of the benefit at all and only get side effects, whether it's the low white counts or bleeding or seizures. So it's really important in parallel to discovering the genetic forms of these conditions to identify the genetic background that may or may not predispose to adverse events. Because you can imagine, and it's been seen in many other conditions, there may be a great drug for a certain percentage of people that never makes it to market because of the adverse events.
Yeah, well, that's what I study a lot. So you should come visit and talk, because we might be able to find markers that would indicate, which patients would suffer from the adverse events, and stratify them out of the treatment, and that could be very beneficial. And I think it's quite possible.
Yeah, absolutely. I totally agree. And this this is a rare disease we're caring about. So we can't afford to be heterogeneous, and we need to empower in the smallest number possible to show efficacy. So if we can eliminate those patients who are going to confound the analysis because of that, we should be able to derive signal from a small group of patients. It will surely be
right, and that's going to be helpful. And then you could get to an early stage, kind of stage one, even almost interventional,
Exactly.
because we would be able to identify potential markers for these adverse events and then measure that, right, as a adjunct to your ongoing study, which
Exactly.
would be great for us.
Yeah, no, that'd be great. And one data set that we have right now that I'd love someone to work on with us is we have patients from the FSGS clinical trial. These about 100 patients who were followed longitudinally, given 2 different drugs, and over time, this closed about 5 or 6 years ago. But just recently, Virginia and our led the study of looking for Mendelian mutations using NIPS, but we've also done genome-wide SNP array. And it'd be really great for someone to overlap the genetic signatures with different outcomes to see if we could start to tease out some of those aspects.
Yeah, great. Please stop by.
Yeah, absolutely.
Thank you.
Thank you.
So Matt, for my curiosity, I want to ask the biology. In preeclampsia for those cases, is this due to the fetal kidney handling salt differently, or is there an immune conflict?
We don't know yet. It's a massive opportunity that will make a big difference in public health. My belief is that it's an immune issue. So we know that APOL1 participates in the trypanosomal lytic factor, so it circulates in a response to interferon and innate immune pathways, antiviral pathways. We think that what's going on in the kidney may be another activation of immune signaling. We know that the placenta has high expression of the APOL1, and it may be that the APOL1 conflict between the fetus and the placenta sets off an immune reaction inflammation, which ultimately leads to bad outcomes. I don't think it's solved.
So I guess in your longitudinal follow-up, you'll be watching for those immune markers constantly.
Yeah, yes, absolutely. In the follow-up, we're collecting samples so that if someone with a massive interest in the immune system can do that kind of work.
Sure. And then the saturation mutagenesis is assayed in cell lines. Do you wish for something that can measure fluid transport, such as something closer to a flow rate?
So I think with I think there's going to be a different assay that's tuned to each specific question. I think for some questions, the cell-based saturation mutagenesis is going to make sense if we have an appropriate readout. But in terms of flow rate, there's certainly going to be some questions that we're going to ask that will be well suited in that way.
So you commented on how CHOP already had a very large pediatric genetic, biobanking. Are you going to eventually compete, or you have a friendly relationship with them?
Yeah, so I only compete with MGI. So us pediatric bio, and I'm just kidding about that, guys, if you don't know me. Us pediatric folks have to stick together. So in fact, I have a long-term relationship with Hauken Arsen, the director of the CHOP Biobank. I just met with him a couple of weeks ago. And what's been interesting is CHOP, Cincinnati Children's, and Boston Children's, which are probably the top 3 children's hospitals in the country, have come together to form something called GRIN, a Genomic Repository Network. And they are it's early days, but they're trying to identify shared wins and shared goals. And I let them know very loudly and vociferously that we'd be very interested in participating from the start. Yeah, so there's already buy-in from this. So it's really exciting time.
Any last question? Okay.
Thank you. Drive safe.